Hello everyone and welcome to my presentation. Today I will be talking about my PhD project, the Electrophysiology of Depression and Chronodisruption. My name is Abdulrahman Sawalma and here it goes. So this project will be in collaboration between INM4 in Forschung Zentrum Jülich and the Palestinian Neuroscience Initiative in Al-Quds University. The supervisors are Dr. Jorgen Damas from INM4 and Dr. Mohammed Hirzallah from the PNI. We will talk about the introduction, then about the master's project, then we go in depth in the PhD project, we go through background definitions, the theory behind the project, study methods and the project plan, and some few nice words about the complementary project between the two collaborating sites. So in general, let's talk about the general theme of my master's project and my PhD project. It's about the study of mood or depression in specific. And depression is affected and affects a number of factors. So it affects brain functionality, it affects the cognitive ability of the patient. It's also characterized by changes in neurotransmitters, serotonin, dopamine, and so on, and circadian rhythm or biological clock rhythmicity. In my master's, I studied some of these factors. So I studied depression symptoms in healthy controls. We also looked at the brain functionality as evident in the EEG theta wave and at serotonin gene 5-HT1A, which represents serotonin activity, and period 2 gene, which represents the biological clock rhythmicity. And in a nutshell, we found that later circadian rhythm is correlated with higher theta power in the brain and with higher depression symptoms. We could not find anything for the genotypes we study. Now, in the master's project was done only in healthy controls. So we studied depressive symptoms in healthy controls. And what we wanted to do is to extend that beyond and to study patients with depression, moving a level upwards. So that is where the PhD project comes in. So it's about the electrophysiology of depression and chronodisruption. And these are the factors that we will be talking about. It's a correlation between depression, brain functionality as measured by electroencephalography, EEG, and magnetoencephalography, MEG, and the cognitive function in the brain, and the chronotype. Of course, we can't start without defining the chronotype. To understand what a chronotype is, we need to start with a biological clock. So, a biological clock, as everyone knows, is the endogenously controlled biological rhythm within the body. And this biological rhythmicity controls the physiological functions as well as the biological functions. Chronotype, on the other hand, it's the variation in this biological clock. In other words, you see people that wake up earlier, they tend to wake up earlier in the day, and they tend to go sleep earlier in the day. And they become more active during the day earlier than average. And these people, they have an early rhythmicity or early biological clock type. This is called earlier chronotype. Later chronotype, sometimes called the night owls or night people. These are the people that wake up late, tend to stay up late at night, and they are called the later chronotype. And most people are somewhere in between. So studying chronotype is important in depression, as we'll see, because chronotype is affected in depression. Now, with that said, we can go into the theory behind the PhD project. First of all, these factors affect each other. First, depression affects brain function. And this appears as changes in information processing and changes in hippocampal function, especially information processing in the hippocampus. Depression also affects chronotype. It appears as sleep disturbances in most people. Up to 70 to 80% have disruption in the sleep-wake cycle, and they complain of insomnia and such sleep problems, and circadian rhythm abnormalities such as changes in the hormonal rhythmicity, physiological functions such as temperature control, and so on. Depression also affects cognition, in which depression affects the responsiveness of the patient to negative stimuli. Now, you see some patients with depression who dwell more on the negative. They only see the negatives around them, and they have less sensitivity to positive feedback. And this is something we'll be studying accompanied by brain function and depression. So subjects would be two types of subjects, healthy controls, who will be recruited from using flyers or using the phone, and patients will be collected and recruited from the psychiatric clinics, either in Palestine or in Germany, and the diagnosis will be determined by a psychiatrist. The modality of choice is MEG. Now, we aim to measure brain function using MEG or EEG in Palestine, and we aim to study the resting state as well as task performance. The modality of choice is MEG or EEG because it provides, both of these methods, as you can see, they provide superior temporal resolution, meaning that we can look at the millisecond resolution. And this is important because this gives better resolution and gives us the ability to look at specific frequencies which are said to be disturbed in depression and in information processing. A few words about the task will be used. So it's a categorization task. The subject is asked to categorize a number of cards into sun or rain, category A and category B. And if the subject answers correctly, they will be rewarded with points. And if they answer incorrectly, they will have negative feedback. And all of this will be done under EEG or MEG to measure their responsiveness and their decision making and how do they respond to reward and negative feedback. The plan in general. So we aim to study the effect of depression on chronotype and cognitive function. This will be correlated with brain function with the task done under the MEG or EEG. And ultimately, we will try to find a response marker. So we will study two types of patients with depression, patients who respond to treatment and patients who fail to respond to treatment. And if we can, we'll find a response marker that can determine response to treatment if it is present. The current state of the project, so we are now in the process of acquiring the ethical approval. We filed for one ethical approval. We got the comments back, so we're now doing the revision. And we also are preparing for data collection, doing a pilot study, so to make sure everything is up and running. Now, while we are doing this ethical approval and reviewing our ethical approval, we are also doing a complementary project. So this project measures the two of the factors that we're interested in: depression symptoms and cognition. And the connection between depression symptoms and cognition is personality, as personality changes in patients with depression. So to do that, we are using data collected in the PNI to study personality in patients with depression. We used a personality questionnaire called the Tridimensional Personality Questionnaire, and as the name suggests, it consists of 3 dimensions, or 3 parts of the personality. These are novelty-seeking, harm avoidance, and reward dependence. This questionnaire has proved to be clinically useful. So what we did is 2 things. So we studied personality differences due to depression. For that, we collected data from 250 subjects, and we tested them before and after treatment, and we checked their personality change due to the presence of depression and due to treatment. Secondly, we tried to validate the structure of the TPQ and tried to test its clinical usefulness. For that, we collected data from 1,000 healthy subjects, and we tried to find the components that make up the TPQ. Now, the TPQ was originally divided into 3 dimensions by the original author, and he did that subjectively. What we try to do is to find the components that make up the data, but we're not subjective doing this. We are objective. We're doing a data-driven categorization using independent component analysis. And we test the clinical usefulness of the original categorization and the new one. What we found is that harm avoidance changes with depression, like follows. So patients have higher harm avoidance at baseline compared to healthy controls. And then after treatment, we find that only those who respond to treatment have lower harm avoidance comparable to healthy controls. And this opens a very nice and lengthy discussion about what comes first. Is it the personality change that predisposes the patient to depression, or is it the depression that causes personality change, which is beyond this very short presentation. Secondly, the second type of result that we got is the test the ability of categorization of the TPQ. So we tested this using two methods. First, we tested the ability to predict the presence of anxiety in our patients of depression, so comorbid anxiety, and the ability to predict response to treatment. And what we found is that the ability to predict anxiety increases to 73% in the new categorization as compared to 62% in the original one. And the ability to predict response to treatment increases slightly to 66% as compared to 62%. And we are working in collaboration between INM and the PNI to publish these results hopefully soon. And with that, I end my talk. Thank you for listening, and I welcome any questions or comments on my email, and thank you very much.